In this video, we will discuss about approaches towards HIV vaccine development. So in the first figure on the left side, and in this figure, the A panel represents how the HIV infect the virus. So let us quickly go through or revise this, how the HIV go through the virus before going on towards the methods or the ways people are trying to develop HIV vaccine. So this is the HIV virus and this is the GP120 on the spike protein, and this GP120 will uh, attach to the cells that has CD4 plus receptor, at the same time also CCR5 uh, co-receptors, and those cells that have these two receptors are prone towards HIV infection, and the uh, infiltration of the HIV virus occurs, and then reverse transcription and integration occurs. Reverse transcription occurs because HIV virus has the single stranded RNA as a genomic material, and it needs to be converted into DNA before integrated into the host DNA. After that, it takes over the cell's machinery, and then transcription and translation occurs. And as we know that everything is present within the virus, that is its envelope, and this its reverse transcription uh, package is also present within the virus. So once it is integrated into the host cell, it 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 will not face much problem afterwards for transcription and translation. And after it is translated in exocytosis, then this new virus can infect the other CD4 plus and CCR5 co-receptor cells. And this is the one of the approach that has been presented here, how to make HIV virus, how to make HIV virus vaccine. So in this approach, these HIV surface proteins from the vaccines will come in will brought in contact with the B cells. In this way, B cells get aware about this kind of proteins, and result in activation of T cells that release interleukins and HIV specific antibodies with the help of plasma cells. In this way, our immune system will get trained against the HIV specific against the HIV specific protein that is usually present on the HIV virus. And the second approach is about using uh, non-pathogenic genes from the HIV virus into the vector, and then transfect it within the humans. At the same time, naked viral RNA is also naked viral RNA is reverse transcriptase, and its non-pathogenic plasmid DNA form of viral gene is also transfected, and these both result in activation of B cells, and B cells, as happened in the previous case, will activate the T cells that release interleukin, and plasma cells uh, that makes the HIV specific antibodies. And this way our body will get trained against the HIV surface protein. And in the other approach, that is macrophage, get activate the CD8 plus T cells, that CD8 plus T cells activate cytotoxic T cells, which will help in apoptosis of this HIV infected cells. And in this second figure is the modern approach that people are now thinking to develop a vaccine, or the patient that are infected with the HIV virus, uh, how they can be cured. So these are the approaches focuses on that part. First one is the stem cell transplant. What happens in this? With the help of apheresis, uh, plasma of the infected patient will be taken and its ex vivo treatment will occur, and by silencing this gene that is integrated into the host cell that is responsible for HIV virus formation, uh, then they can again transplant these cells again into the patient. And in another case, uh, what is the allogenic case? Autologous case is using the patient stem cells. Allogenic case is using other person's stem cells. In this case, the CCR5 modified cells, as we have discussed in previous one of the previous videos, that CCR5 can be modified so that these cells become HIV resistant. So from the healthy donor cells uh, that have CCR modified in it can be used with the help of stem cell transplant to transfect into this infected patient, and with time this transplant cell will take over the whole body, and the HIV infected cells will vanish with time. And in this another approach is about the CRISPR-Cas9 editing. In this case, CRISPR-Cas9 system can be used to edit or to remove or to deactivate the gene that is responsible for HIV virus translation or replication. So this gene editing can be done by changing the shift by changing or shifting the base pair that results in formation of a different final product or proteins or sequence of amino acid, and does not perform functions that is being performed by HIV virus. The third one is the shock method. In this, antiretroviral therapy is being used to kill the cells that are HIV HIV that have HIV-1 provirus. So in this case, the antiretroviral therapy will induce apoptosis, and it will activate CD8 plus T cells that will lyse the HIV-1 provirus. And this reversing agent will also be used so that this HIV-1 provirus, this kind of cells will not form within the patient in within or recently, will not form within the patient in a short duration of time. And third one is the block strategy. In this, ART is not used, that is antiretroviral therapy is not used. In this case, what is been used is some latency promoting agent is being used that will silence the gene that is responsible for producing HIV virus, or that is been integrated into the host cells. So these are the some of the promising latency promoting agents mentioned here. And in this way the host integrated HIV virus will be logged, and or we can say there is a epigenetic silencing that can be done in this block strategy. So these are the few strategies by some of which are to develop HIV vaccines, and some of which are to treat HIV infected patient. So that's all for this video. Thank you for your time.